Well, good day to everyone joining us and welcome to today's X-Talks webinar. Today's talk is entitled Comparability of 5 Alternative AAV9 Downstream Processes from Ultracentrifugation to Chromatography. My name is Ryan Muse and I'll be your X-Talks host for today. Today's webinar will run for approximately 60 minutes, and this presentation includes a Q&A session with our speakers. Now, the webinar is designed to be interactive, and webinars work best when you're involved. So please feel free to submit your questions and comments for our speakers throughout the presentation using the questions chat box, and we'll try to attend to your questions during the Q&A session. This chat box is located in the control panel, which is on the right-hand side of your screen. And if you require any assistance along the way, you can contact me at any time by sending a message using the same chat panel. At this time, know that all participants are in listen-only mode, and please note that the event will be recorded and made available for streaming on xtalks.com. At this point, I'd like to introduce our speakers for today's event. Alesh is the Executive Managing Director of Sartorius since its founding in 1998. Alesh is one of the main inventors of the CIM convective interaction media monolithic columns technology and the co-inventor of many analytical methods, including pDNA, mRNA, AAV, and other viruses. Joe Ballidier is the Senior Vice President of Research and Development for Advanced Medicine Partners. Joe has 29 years of biopharmaceutical experience with 8 years of AAV-based gene therapy experience. Joe developed the upstream process for Zolgensma while at the Avexis Novartis and led the process development efforts at Jaguar Gene Therapy. And Mike Hatfield is the Senior Director of Research and Development at Advanced Medicine Partners and former scientific lead of downstream process development for Zolgensma at Avexis Novartis and AAV Therapeutics at Jaguar Gene Therapy. Mike has worked in the biopharmaceutical industry for 32 years, including 14 years with oncolytic viruses and 10 years with viral gene therapy. But now, without further ado, I'd like to go ahead and hand the mic over to our first speaker for today. Alesh, you may begin when you're ready.
Hey, thanks, Ryan. So can you just go next slide? So before starting with the company presentation, I would just really like to thank really a number of scientists and developers with AMP and via separations to put this enormous work together. There has been really a lot of work behind, a lot of efforts, took a lot of time. So thank you to everybody contributing to this work. And then really shortly about Sartorius via separations. So we are part of Sartorius, kind of a gene therapy core of Sartorius. And our main activities would be more into the downstream processing. We also do some upstream. First of all, we are known for chromatographic columns based on the monoliths, which you can see on the left side here on the slide. So being developed almost 30 years ago, really dedicated to work with viral particles, with nucleic acids, really purpose-designed chromatographic media with advantages such as better resolution, better capacity, and also better recoveries when it comes to the viral particle and also nucleic acid separation purification. The columns starts with 1 ml, goes up to the 40 liter. We are bringing new formats to the market, the HR, high reproducibility. The aim is to really have batch-to-batch, scale-to-scale column, really extremely reproducible, tested with the AAV when it comes to the adeno-associated virus purification and released by the separation of the AAV. Of course, we offer 96 well plate formats, 24 well plate formats as kind of screening tools. Analytical columns, always just the same monolith in for fast analytics and also fast analytical systems, which allows us, I would say, in the first place, to perform much faster process development, and then secondly, to use it as an in-process control. Of course, they can be used also for a product release. Last but not least are our cornerstone processes. And next slide, please, Ryan. Where we would have a possibility to help our customers with the process development. Can be just kind of small feasibility study, or we can do everything from the process development and tech transfer to the CMO or to the sponsor site. So far, we tech transferred more than 60 processes, full processes, basically coming from the plasmid DNA of any size, mini circle DNA, doggy bone DNA, different RNA molecules, adenovirus particles, adeno-associated viral particles, where we tested or worked with more than 20 different serotypes so far, flu, vaccinia, exosomes, bacteriophages. So it's really about the viral particles. And we go next slide, please. Yeah, we took part also in the process development at the time with Mike especially and Joe to bring the Zolgensma to the market. I would say that this was really, there was a need to do the process development really very fast to help kids as soon as possible. And for this, you really need quite some experience, but also fast analytical tools to help you with the process development to enable faster process development. And with our in-process PET-fix tools, this can be possible, would be possible. And the next slide. Yeah, I will just shortly touch the process which we would be providing, or which we would advise to our customers to work with when it comes to the AAV, but more deep dive in will be presented by Mike. So the harvest, then we can perform detergent or salt lysis. We prefer salt lysis because in some cases we may reduce the amount of chromatins being formed. The next step would then be chromatin removal. Can be done by flocculation, TFF, DNase treatment. Solid phase extraction is something we are bringing now to the market. If there is a good expression, we would just go to the capturing column, which is in our case cation exchanger. We believe that cation exchanger has several advantages when compared to affinity approach. This is why we are still kind of pushing for this solution, especially when it comes also to the pricing. And you also see from the Mike's presentation later on that there are some other advantages. And then we get to the final polishing steps. For this, we have 3 different ligands. Nowadays, I would really go for QA-HR, so high reproducibility columns, which to my opinion would perform for most of the cases. And worth to mention, not only to get rid of empties, but also partials, and also get rid of endotoxins. And we also have some data about viral clearance of using this column. And with this, let me pass the word to Joe. Joe, please go ahead.
Yeah, thank you, Alish, for that overview of Sartorius. And thanks everyone, first of all, for the attendance on the call today. Appreciate you being here with us. And I would like to take a few minutes to give you an overview of Advanced Medicine Partners, or AMP as we like to call ourselves. Moving to the next slide. So AMP, or Advanced Medicine Partners, was officially formed in January of this year. So although we've only been an official company for a short time, our team of 50 employees has been together for many, many years in this industry. And many of our core team was directly responsible for the development of the manufacturing process, the analytics, and also for the global commercialization of Zolgensma. So with that expertise in mind, we're new as a company, but long on experience in this field. And so one of the philosophy differences of AMP that you may see where we're different is we don't view as a service provider that an IND is the goal. We view that as a milestone on the way to the goal to commercialization. So we're employing a lot of the lessons we learned with that commercialization of Zolgensma in our approach to how we support clients with development of their own programs. So with this philosophy, we've been able to, develop a platform and analytics that are very robust. And at AMP, we don't believe that you should have a trade-off between purity and yield. So at AMP, we insist that our clients get both. And so today we're here to talk about with Mike how we achieve high purity and high yield at scale for our clients. And also we anticipate future needs at AMP. And so we'll give you a brief overview of our expansion projects that are in place and how we plan to grow over the next few years to meet, emerging demands in the AAV gene therapy replacement field. Next slide, please. So highlighting our manufacturing capabilities currently, we've run many batches with our platform process. We supplied multiple preclinical studies for clients and for Jaguar Gene Therapy. And we know that our process is scalable from 250 milliliters all the way to 500 liters without compromising product quality or yield. We've demonstrated our process over multiple serotypes as well as engineered capsids. So our process has great utility in this respect. And we also understand that payload within an AAV does matter. So we have a lot of experience with multiple genes of interest, and you can appreciate differences between self-complementary and single-stranded DNA. But however, we notice that subtle differences such as promoter changes and other things and how they can impact a process. So we're able to modulate our spine of our platform process to meet these high purity and high yield needs for any gene of interest for any program that may come our way. Our process is very robust and no exotic materials, no equipment needs specific to our process. It translates very well, and we have successfully transferred our process to 2 different manufacturers with 100% manufacturing success rate. So for many years, our team has been on the other side of the table as a client with CDMOs. So we have a unique experience that we understand what clients need, as well as, what access to our facility process and most importantly, information that they may need. And we can customize and be flexible with any client on meeting their expectations for the services we provide for them. And with expanding into the future, we are currently constructing the AMP flagship facility in Durham, North Carolina. And this facility will have 174,000 square feet. So we can easily meet the needs that we have in 2025, but also we have enough room to grow with our client needs to meet future demands seamlessly and without impact the timelines. Next slide, please. So highlighting our existing capabilities today, we have 2 process science laboratories located in Cary, North Carolina. We can run our 250 ml Amber systems at the scale, and these are really our workhorse for doing early feasibility or manufacturability assessments for clients, as well as performing large-scale batches up to 1,000 liters. So we know every program has its own specific needs, from ultra-rare programs to more common disease disorders. And so that's why we've developed a portfolio of processing scales and equipment to accommodate 50-liter batch sizes all the way to 1,000-liter batch sizes, depending on specific program needs. So this is available today and fully operational. To highlight our analytical testing facility in Durham, North Carolina, one of the lessons we learned with Zolgensma is you cannot invest enough early in analytical capabilities to understand your process and its quality attributes. So we've invested substantially in analytical method development, and we have multiple methods in-house that are available for process characterization as well as GMP testing. One assay to highlight here would be our capability in potency, in cell potency. So what we found over the years, even though it's not a requirement to have a potency assay necessarily at IND stage, we know that that's key to characterize your process for any process changes or for impacts of scale up. So instead of waiting until BLA timeline to develop a potency assay, we do this early, and we feel that we have a best-in-class team that can do cell-based potency today for any of our client needs. Next slide, please. And in closing, this is, an artist rendition of our facility in Durham, North Carolina. It is on track to be fully operational in Q1 of 2025. And fortunately for many of us, The material and equipment supply challenges associated with the pandemic are long behind us, and our project team has met consistently every milestone with this project, and we are substantially confident that we will meet this timeline and be ready. And I'm personally looking forward to being co-located with all the AMP operation folks in one location, and we look forward to potentially hosting some of the folks on this call to visit to the site and working with you in the future. So with that, I'd like to hand over to Mike to talk about the reason we're all here today and to discuss some of AMP's process science.
Okay, are we on there? Yep, there we go. Okay, good morning. Thank you, Joe, for that background for AMP, and I'd like to thank Xtalks for this opportunity to discuss our AAV9 process comparability study. As Alish mentioned, it took a lot of people and a lot of time to get all of this done. Next slide, here we go. So by way of background, the AE vectors have proven to be very useful in treating a range of monogenic disorders such as SMA with Zolgensma. We believe that designing a manufacturing process that emphasizes product quality and safety with extensive characterization of the process and product should be prioritized. We do this early in our development rather than waiting until we have early phase clinical phase results. We do this with improved analytical methods from the last several years that have aided in better characterizing the impurity profiles of the process and the products. Identifying and enriching functional capsids is critical for the intended therapeutic benefit. Oops. Got this little box covering up part of my slides. Okay, well, I'll work from the hard copy. Anyway, ultracentrifugation has long been the gold standard for separation of full capsids from empty and partial capsids. And today we present data comparing full capsid enrichment yield and impurity profiles for drug substances resulting from 4 different 2-column processes to the UC gold standard. Study objective, again, is to provide an extensive comparability analysis of 5 different downstream processes, including the UC-based process and 4 2-column processes. And here's sort of the abbreviated process flows to discuss. First thing, though, is that keep in mind that all of the lysate used for these studies came from one single 500-liter bioreactor run. 200 liters were used for our standard UC process run, and then 50 liters were used for each of the 2-column process runs. Initially, you have the lysate. It's a surfactant released lysate. We either flocculate to load the CEX, or we use a clarification TFF system to generate the load for the affinity pH step captures. Going down through there, the capture is either loading the flocculate clarified floc onto the CEX column, then eluting with a sodium chloride gradient for processes 1 and 2. For process 3 and 4, we load the same CEX column, but we elute with a pH gradient, giving more details later. And finally, in process 5, it's using an affinity resin that we elute with the step gradient. In most cases, we use TFF to concentrate the material before going on to the full capsid enrichment AEX column. However, for the UC process, number 1, we found that we had to use a AEX filter in flow-through mode to remove a couple of identifiable contaminants that the CEX and the UC did not remove. When you're using AEX in a bind and elute mode, we don't see this issue. So if you have a salt gradient, we definitely need to do a TFF before loading the AEX column, because the AEX column requires very little salt to knock off the capsids. Nice thing about the CEX-PEH gradient method is we are at low salt at the point we do the pH shift, and as such, we don't have to do a TFF to remove that salt. For AEX, well, again, UC I'll describe in more detail on another slide later. For the AEX, it's load the AEX column and a sodium chloride gradient for all 4 of the for runs 2 through 5. One interesting twist was process 4. We actually take the AEX LU8 from process 3, split it, half of it went to DS by TFF, the other half we diluted 8-fold and then reloaded on the same column, and we're able to increase the purity as well as both host cell protein, those sorts of impurities, and process impurities. More details. So why do we use flocculation for CEX? Flocculation improves column capacity and step yield by removal of the host cell proteins as well as plasmid DNA and host cell DNA impurities. All of this really is in the, not all, but a major component of this impurity profile is in the form of chromatin. This binds to the these negatively charged components. Sorry, the positive charged components bind to the negative binding sites on the cation exchange column. And the removal of the chromatin greatly improves our chromatographic capabilities. Chromatin from lice cells is a complex of histones, DNA, RNA, and host cell proteins to form these nucleosome multimer complexes. Nucleosomes can be thought of as sort of tuna can shaped things with a highly positive surface on the outside of the can and a highly hydrophobic surface on either lid. As such, you end up having highly positive sections on the histone parts. You've got the DNA attached to that. Tails of DNA are highly negative, and then you have those hydrophobic surfaces. So pretty much any column you stick it on, CEX, AEX, or hydrophobic interaction, this material will stick to it and thereby add more material that gets a different set of binding sites. So you turn just about any column you apply this crude lysate to, you will make a multimodal column out of it. Based on that, anything you can do to remove this chromatin and these other impurities increases not only our binding capacity, but also the specificity at which the column can run. Clarified floc volume is about 2 times greater than the starting lysate volume. So we go from 50 liters to 100 liters. Because we use the 6 micron channel monoliths, we can flow at 7 CVs per minute, meaning that we can load 100 liters of clarified floc in about 35 minutes across a 400 mil column with these 6 micron channels. Conversely, if you're working with the affinity, there isn't the need for this removal of process impurities because the affinity column has this selectivity of the affinity ligand. Therefore, that's not as much of a concern. However, column capacity is dependent on the residence times with these bead-based affinity resins, and loading flow rates are limited to about 0.3 CVs per minute by this residence time requirement and column back pressure. So in order to get the loading in a reasonable period of time, we use TFF to concentrate the material about 7-fold. And when loading on 165 mil affinity column, as we did in this study, we were able to accomplish the load in 3 hours compared to 20 hours if we hadn't done that concentration step. Going back through these basic processes again, You've got the capture for process flows 1 and 2 is on the SO3 column. The capsids are eluted with a sodium chloride gradient from 500 to 2000 millimolar salt over 20 CVs. Processes 3 and 4, we load on the same CEX column. We elute with a pH gradient. Again, this column is 400 mils, 6 micron channeled SO3. The gradient elution after we have rinsed the column with the loading buffer without salt, we can elute from pH 3.5 to 9.3 over 20 CVs. Then for capsid the process flow 5, which is the affinity, we take the TFF retentate, load that onto this column, and elute the capsids with a step elution from pH 7.5 to 3.0. Full capsid enrichment by ultracentrifugation. I'll talk a little more detail next slide. For process flows 2 to 5, we're talking AEX chromatography, sodium chloride gradient. We load again a 400 mil 2 micron channel. That's not millimeters, by the way, that's microns, with a QA column, and the gradient, it's a shallow gradient running from 12 to 132 millimolar sodium chloride in 30 CVs. As I mentioned a little bit ago, for process 4, we took the LU8 from process 3, split it, half of it was then diluted 8-fold and rerun on the column. Here's the ultracentrifugation, and let me get my mouse out. We performed this at the 200 liter scale. The TFF was buffered from the flow through from the AEX filter, was buffer exchanged into the 3 molar cesium chloride buffer. That buffer exchange concentrated material was loaded in 40 mil tubes. These were spun in a VTI 50.1 vertical rotor for 9 hours at 50,000 RPM. As you can see over here, the empties are up at this place, this portion of the gradient. Partials are these 2 minor bands here. This is the product band down there. And there's a high density, a faint high density band down here deeper in the cesium chloride gradient. So the bands were collected manually by puncturing each tube with a needle and withdrawing the product bands. Product bands from multiple tubes were pooled for TFF processing to drug substance. Here are the analytical methods for the comparability study. Most of them fairly routine. Vector yield, genomic titer by dt-PCR, total AAV titer by ELISA, host cell protein by ELISA, plasmid DNA, a CANR amplicon by dt-PCR, host cell DNA by a qPCR kit. Empty full partial content we assessed by both AUC, CDMS, and mass spectrometry, all sort of orthogonal methods for that assessment. Aggregation by size exclusion chromatography, protein purity by capillary electrophoresis, capsid protein charge heterogeneity, very interesting assay we'll talk about in more detail in a moment, by image capillary electrophoresis focusing, image capillary isoelectric focusing. Intact capsid charge heterogeneity by capillary isoelectric focusing, short read NGS by ONT, and long read NGS by PacBio. Finally, the functional assay by in vitro transduction, followed by monitoring the fluorescence of the GFP protein expression. Here is that charge heterogeneity section. In this situation, when you're looking at stability, any changes in charge heterogeneity really end up being deamidation. Deamidation is an intermolecular reaction that depends primarily on time, temperature, and local backbone flexibility. Glycine residues of asparagine that are followed by glycine are the most likely and most rapidly deamidated. Each deamidation produces an additional negative charge on the capsid at physiological pH and can strongly decrease AAV potency. Up in this upper left corner, you can see the biochemistry, or the chemistry, I guess you might look at it. Basically, this amine on the protein backbone attacks the carbonyl on the asparagine side group, releases an ammonia, forming a succinate intermediate, a succinamide intermediate that then hydrolyzes to form either aspartic acid or isoaspartic acid. Down here in this lower left corner, you see the ICIEF plot from a forced degradation study. The amount of material in the different peaks is monitored by fluorescence of tryptophan, endogenous fluorescence of the protein. And the black line comes from the T0, or the material that was just thawed and applied to the assay. The red trace here comes from the material that was stressed for 5 days at 40 degrees C. As you can see, well, first off, let's talk about this. You spike the samples with these protein controls, one of which has a PI of 5.85, the other one has a PI of 7.05. These allow us to basically standardize where these guys are coming up, these fragments are coming up on the electrophoresis. And the full native VP3 here, VP3, native VP3, you see a reduction from time 0 to 5 days at 50 degrees C, and the corresponding increase in these deamidated VP3 variants. In this plot over here, you see time versus the amount of amidated, or basically you're looking at the loss of asparagines with time. Over 5 days for here are out to 56 days at 25 degrees C. And basically nothing if you keep material frozen for that period of time. This correlates quite nicely with what you see in the potency assay. So this is potency versus time at 80 degree, minus 80, 25, or 40 degrees C. So the correlation between the deamidation and the loss of potency, I believe, is exceedingly striking. And I would contend that this ICIEF method appears to be, it's the best potency assay, not potency assay, the best potency indicating assay we have short of the true potency assay. So it's a stability indicating assay for potency. Now to the data itself, we're looking at the vector genome recovery from lysate all the way over to the drug substance. And what we see is that the 2-column CEX, AEX processes come in with yields from 54 to 60%. Our UC process gave a yield of about 33%. That's within ballpark of normal, maybe ranges 30 to 40%. In our hands, in this first large scale run with affinity, we ended up with 24% recovery from lysate all the way to drug substance. In discussions at a poster at ASGCT in Baltimore, the folks who do affinity on a regular basis said that their yields, cumulative yields, are more like 30 to 40%. So with our process coming in at 54 to 60% recoveries, we have anywhere from 20 to 100% more doses for patients with these 2-column processes. Next, we're going to look at process-related impurities in the drug substance. Note, all of this data is from the drug substance, unless stated otherwise. And we see a trend here where processes 1, 4, and 5 demonstrate lower residual process impurities. Note that the host cell protein impurities are below level of quantitation of 4 nanograms per mil. Next slide, we've got, this is looking at vector purity by CESDS. It's sort of like SDS page, where you're looking at all of these guys are running 99% recovery, 99% purity, plus or minus 0.5%. You look at aggregation, process 2 and 5 have a little more aggregate out here in the plots, is demonstrated by this overlay. Is this biologically significant? Probably not, but there is a slight difference there. Again, this trend of process 1, 4, and 5 producing the best quality profile. You can see that the empties are quite low in the UC. This is actually a little lower than we sometimes see for the UC, but it does happen. Process 4 and 5, you've got 2% empty capsids, and in all these cases, you've got full capsids in that 93 to 94% for those 3. It's lower for a couple of these other processes 2 and 3. Of note, of the 3 orthogonal methods, AUC, mass photometry, and CDMS, the rank order for empties and full capsids are the same. Partials are a little more, they're not always the same rank order. I attribute this to somewhat of a lack of resolution in the mass photometry and the way you have to drop the integration lines. That's also somewhat true of the CDMS when you do this in a 2-dimensional plot. Now we're looking at vector purity of the drug substance using DNA reads. Keep in mind that short read NGS, because you cut up the DNA as you're forming the library, is more of a mass assessment of, and the mass of DNA that comes from which initial sources, be it the gene of interest plasmid, backbone, the rep cap, the P-help, or host cell DNA. And as you can see, we've got 95 to 97% of the mass of DNA that's encapsulated in these proteins actually comes from the gene of interest part of that plasmid. The next level contaminant, or the highest contaminant, comes from the backbone. Minimal rep cap and help. Host cell DNA, there might be a difference between these CEX-PH and affinity versus the CEX sodium chloride up here on the top. Again, is that significant? Hard to say without replicates. Then we go to the long read NGS, and this is one of my favorite parts. You know, long read NGS indicates that 84 to 87% of the encapsulated DNA was full-length ITR to ITR functional genome capable of expressing the gene of interest, EGFP. Yeah, we had by mass, you know, almost 95% of material here, but we see by full sequencing that some of that mass is actually partial genomes. It's pretty amazing what the cells do with this DNA, the way they can categorize and make self-comp components out of what should be a single-stranded DNA. They merge things together. But we see in general, you got partial genomes being the largest contaminant, followed by backbone. And then when it comes to P-help, host cell DNA, and chimera, they're all below 1%. Finally, we're looking at the deamidation and potency measures. We see no differences in the PI of the main peak. The acidic forms are all within variability of the assay. Main peaks in the same spots. Basic forms, of course, are not changing. The intact PI, or the PI of the intact capsids are the same. When they ran that stability study at 5 days, the PI of the intact capsids was, let me look that up. At 5 days, it was 6.4, and at 3 days, at 40 degrees C, it was 6.9. So, and in that study, the control, it was PI of 7.2. So we're not seeing an issue even in the intact capsid PI. Potency, however, the 4 2-column processes appear to be a bit more potent than the UC. This was a surprise. However, to support that, the UC drug substance was used as control on all of these plates when you were comparing the 2-column processes. So in every plate, we see the UC being 10 to 15% lower potency than for the 2-column processes. Again, you need replicates of different runs to really prove this point, but it's definitely suggestive of that. So conclusions, yields from the 2-column CEX, AEX processes were 1.7 to 2.5% greater than those seen with the affinity process and the UC process, respectively, UC, affinity. Full capsid enrichment, all but one of the 2-column processes produced 90 to 93% full capsids at drug substance, compared to 94% for the UC process. For the quality attributes, 84 to 87% of the capsulated DNA in the drug substances were full-length expression cassettes capable of expressing the EGF based on long-read NGS. The UC CEXPH with 2-cycle QA and the affinity QA processes produce the best process profiles with respect to host cell DNA, empty full capsids, and host cell protein. Potencies of the drug substances from 2-column processes were equivalent and potentially greater than that of the UC purified drug substance. Scalability, these 2-column processes can be readily scaled to 500 liter and larger batches. Trying to figure out how to get this out of my view. Sorry, folks. Moment. There we go. So, from the AMP perspective, we presented a comprehensive characterization of 5 downstream AAV9 processes at scale in terms of full capsid enrichment, yield, and residual impurities. Overall capsid enrichment and efficiencies at upstream influence the success of downstream operations. These include optimizing transfection conditions, production duration, target cell densities at the upstream steps, Design of the gene of interest construct certainly impacts the partial capsid content. Critical quality attributes of drug substances resulting from two-column purifications were comparable to the gold standard UC process. Manufacturing platform should allow for variations in process inputs, including serotypes, engineered capsids, and DNA constructs. AMP is well equipped to run both UC and two-column manufacturing processes at scale in both PD and GMP environments per client program needs and product lifecycle. AMP processes are aptly supported by best-in-class advanced analytics, including a suite of orthogonal methods, 58 internal and 19 external, to fully characterize the process and product. Our process science and analytical GMP testing facilities are operational in North Carolina today and can support IND enabling work. AMP's clinical GMP manufacturing facility is on track to be operational in Q1 2025, as Joe mentioned a few minutes ago. To learn more about us, please visit the site at ampgtx.com. For webinar-related questions, you can contact Joe or myself. Discuss how AMP can de-risk your CMC work and expedite your path to IND submission. Contact Rob Mancino or come in through the partnership link there listed. Acknowledgements. As Alish said at the outset, this was a huge amount of work over actually, we're up to about 8 years total together, working with the BIA process development group and the bioanalytical group at BIA. This work, small scale studies, feasibility, et cetera, were performed at BIA. The large scale work came over to AMP, and kudos to the AMP process development group who did this with minimal tech transfer and virtually no scale up from 1 liter up to 50 liter two-column process runs. Can't say enough about the AMP analytical group. Will leads that. Mike Murphy is bioanalytics, the bioassay. We've discussed Austin's biophysics with the deamidation. Steve Alassi has worked tirelessly to sort out how to do the short read and long read NGS assessments. Again, my main leads over the year were Maya, Rock, and Moitza, and Alish, and could not have done this without their insights over all these years. With that, I'll finish and we can have questions.
Thank you very much for this very insightful presentation from everyone. I'd like to invite our audience to continue to send their questions or comments right now using the questions window for this Q&A portion of our webinar. Now, I've already received some questions for our group, so we'll get ourselves started with those. The very first question, and Mike, I think this is best directed for yourself. Someone's wondering, how was the AAV functionality affected by the CEX run at pH 3.5, and how long was the AAV approximately exposed to pH 3.5 during processing?
Good questions. When we're flocking, we drop the pH to 3.5 for probably about a half hour before we start the filtration. The filtration takes another hour, then we load, we elute. I'd say total time at pH 3.5 is probably about 3 hours. And then it's neutralized to pH 8.0 prior to moving forward. In the case of the pH elution, however, you got about the same length of time at 3.5, but we do elute and it comes off the column at about 8.1 in this particular study. So direct answer, I guess, is about 3 hours at pH 3.5. We've done a study a long time ago where we tested a range of pHs and where you lose bioactivity, where you lose, ddPCR signal, and it took quite a few hours at 3.5. There seems to be a pretty good cliff shortly after 3.5. By the time you get to 3, it starts to drop a little faster at 2.5. As all the guys who do affinity know, you better neutralize that ASAP. So.
Wonderful. Okay, thank you so much for that answer. I've got a question here that came in a little earlier for Alesh. It's curious about which chemistry you think would work best for the capsids separation.
I would first suggest the QA HR to be tested because this is, to my opinion, working for the most of serotypes. If this would not work properly, then I would also test the Prima T and Prima S, but I would always start with the QA. And only if the QA is not providing the separation good enough, I would pick the other two, the Prima S and the Prima T. Especially Prima T with maybe AAV9 would be always good to test together with the QA. But in all other cases, the QA would be my favorite.
Perfect. All right. Thank you very much. The next question here is another one for you, Mike. Someone's wondering about what's your understanding of applying the low pH fluorication, approach to other common serotypes in terms of stability?
We have really not seen an issue with this 3.5 for, again, those 3 hours, and we've done serotypes 5, 6, 8, and 9. 2, we had some difficulties, but that really appeared to be more in the aggregation and before we even got to flocculation. We're in the process of working on that at this point. But various serotypes, we've also done some modified capsids and have not found a big issue here.
All right, very good. Thank you. Another question for you here. Someone's curious, how reproducible is the pH gradient on the CIM SO3 column?
It's incredibly reproducible. We've been doing this for years at 200 mil, 800 mil, 50 liter, 200 liter, the gradient, and because it's mass transfer system, you don't have flow rate issues. We can run at small scale. We've run flow rates up to 10, 20 CVs a minute. As the columns get bigger, you get a little limitation there, but even if it's a 6 micron channels, we're running 7 CVs per minute. The gradients run beautifully. The only place we've seen an issue with gradients is on the AEX on large skids. They sometimes don't like to run at 0% B up to 100% B. And so that's why we had the 32, I think it was 12 millimolar up to 132 millimolar, because that was basically setting up the start of that gradient at 9% B up to 100% B. So that's more of a skid function than a column problem. And the columns are very reproducible.
All right, very good. Thank you. Next question here is curious for capsid capture. Why choose CEX over affinity chromatography?
A couple of things come up. One, in our hands, it's quicker, and with the pH gradient, we believe we can get this process all the way from lysate to drug substance in 2 days without a whole lot of difficulty. The other part is that the affinity, has been designed for all the basic serotypes. We've talked to folks at ASGCT, and there's a number of folks who've said that it either won't stick to the PAN's the PAN affinity column, or sometimes they stick and they can't get them back off. So it could be a bit of a case by case, but for us, we find that the CEX with proper removal of these contaminants prior to hitting the column gives us a lot more flexibility, and we believe it's going to be very useful, particularly for these modified capsids.
Okay, very good. Now, another question here is curious, why choose a 2-column process over UC?
Well, it's certainly a longer process. With the UC, you have a 9-hour run. You have to manually pull these. Well, we're working on ways not to manually pull, but currently we manually pull the bands. This takes 4 days easily, and it's tedious. Centrifuges, we have occasional issues with a centrifuge deciding to stop partway through, and that's a whole bunch of material, but that gets ironed out and works out well overall. But we just find the yield greater, and to our surprise, I have to admit, from this study, to our surprise, the critical quality attributes look good with the 2-column processes, particularly the 2 we favor, the 2-cycle QA and the affinity. Both of those give us quality comparable to what we see with UC, without all of the tediousness of the UC tubes and the spins.
All right, fantastic. The next here is wondering what flocculant do you recommend prior to CEX processing?
I wish I could tell you more. Lawyers have suggested I be a little quieter on that for the moment. We do use anion surfactant, and we use a PEG to help solidify that for the clarification steps. I can say that much.
Very good. Thank you very much. Another question here, someone wondering, do you think the capsid demediation assay will become an industry-wide stability indicating assay?
I would think so. I mean, our work has really shown that with a couple other actually clinically viable plasmids or constructs. It's not just EGFP that we see this correlation. And I think the precision of this assay is apt to be better than your typical bioassay. I mean, our bioassay group does phenomenal work. It's amazing to see the CVs on those assays, but most bioassays just don't have that sort of level of precision. And I think this assay, it's not going to supplant a bioassay. The FDA is never going to let you get away with that. But as far as tracking and monitoring your work over time, I think this assay is really excellent.
All right, wonderful. Well, thank you very much for that answer and for all of the answers today. However, we have reached the end of the Q&A portion of our webinar. If we couldn't attend to your questions, know that the team at Sartorius and AMP will follow up with you, or if you have some further questions, you can direct them to the email addresses or visit the website that's up on the screen. I want to thank everyone for participating in today's webinar. You will be receiving a follow-up email from XTalks with access to the recorded archive for this event. A survey window will be popping up on your screen as you exit, and your participation is appreciated as it helps us to improve our webinars. Additionally, I've shared a link to view the recording of today's event in the chat box, which you can also share with your colleagues so they may register for the recording here as well. Now, please join me once more in thanking all of our speakers for their wonderful time here today. We hope that you found the webinar informative. Have a great day, everyone, and thank you for coming.
Thank you. Bye-bye.
Thank you, bye.